for interpopulation improvement. Which you will see for interpopulation improvement. So let's get back then to recurrent selection. And say a few things about recurrent selection. Of course, we will be mentioning it the other way when we get into intrapopulation improvement. But let's just say a few things. I said we will get into details on this, so I didn't cover it in much details when we were looking at self-pollinated crop species. Recurrent selection, the initial idea of this approach to selection was brought by Hayes and Garber in 1919. And then later we had East and Jones in 1920. And then another group of scientists worked later, which was Hull in 1945. So they defined recurrent selection as reselection generation after generation with intermating of the selected plants to produce the population for the next cycle of selection. Defined it as reselection generation after generation with intermating of the selected plants to produce populations for the next cycle of selection. And the aim of this method being to help isolate superior inbreds for the population subjected to recurrent selection. The idea was to identify an outstanding inbred line. The idea of identification of an outstanding inbred line depends on two factors. One, depends on the proportion of superior genotypes present in the base population from which the line is isolated, and it also depends on the effectiveness of selection during the inbreeding of desirable genes. Okay. So what are the key features of this recurrent selection? The general key features is that a recurrent selection cycle consists of three main phases. The first phase, individual families are created for evaluation. In the first phase, individual families are created for evaluation. Then parents are crossed in all possible combinations. In the second phase, evaluation of the plants or families or selections of a new set of parents. And in the third phase, selected parents are intermated to produce population for the next cycle of selection. So that this pattern or cycle is repeated several times. Normally, it is repeated 3 to 5 times. The first, you have the original cycle, which is called cycle 0, and is called the base population, while in the subsequent cycles, which are generally named as cycle 1, cycle 2, up to n, you have differences you can see there. So the subsequent cycles, of course, cycle 1, cycle 2. So those are the things. So each cycle, of course, cycle 0, you have 1, 2, 3. Then it is repeated again in cycle 1. It is repeated again in cycle 2, and so on and so forth, as you will be able to remember. Where is recurrent selection applied? Recurrent selection may be used to establish a broad genetic base in a breeding program. Because of multiple opportunities for intermating, the breeder may add new germplasm during the procedure when genetic base of a population is rapidly growing narrow. Okay? So when the breeder thinks that their genetic base is growing narrow or variability is reducing, may decide to employ this approach. Recurrent selection is superior to classical breeding when linkage disequilibrium exists. In fact, the procedure is even more effective when epistatic interactions enhance selective advantage of the new recombinants. So recurrent selection may also be applied in legumes like peanuts and soybean. It has also been applied in barley and oats. So that was about applications, where it is applied. Genetically, what is happening when we do recurrent selection? Recurrent selection schemes exploit what we call additive, partial dominance, and dominance. Sometimes the gene action exploited here. One is additive, two, partial dominance or overdominance. And where testers are not used, where testers called simple recurrent selection. Selection for general or specific combining ability is applicable, and therefore we are exploiting other gene effects like dominance, overdominance. So we have recurrent selection for general combining ability, which is more effective when there is additive gene effects. Then we have recurrent selection for specific combining ability, and so on and so forth. So all the three becomes important in terms of genetics. I will get back to these genetic issues when I would have covered some mathematical concept with you, but I want to still mention that there are several things that happen in recurrent selection. In recurrent selection, we have opportunity to control the parent. So genetic control over both parents will double when we use this approach of recurrent selection. And we can control these parents in several ways. We can self the selected individuals. We can select before pollination, or we can do recombination. We can wait for recombination to occur among the selected clones. So we have there's control of the parents in this type of selection. So there are several types of recurrent selection. There are basically 4 different types of recurrent selection schemes. We have what is called simple recurrent selection for general combining ability, recurrent selection for specific combining ability, and we have reciprocal recurrent selection. Simple recurrent selection is similar to mass selection. Simple recurrent selection is usually similar to mass selection in its first 2 cycles, and it is usually 1 or 2 cycles per year. Sorry, 1 or 2 years per cycle, sorry. So the procedure does not involve use of a tester, and selection is usually based on phenotypic scores. It's based on what you see. This procedure, simple recurrent selection, is sometimes called phenotypic recurrent selection. Recurrent selection for general combining ability, we will see this when we discuss half-sieve progeny selection. It's basically a half-sieve progeny test procedure in which a wide genetic based cultivar is used as a tester. A wide genetic based cultivar will be used. The is evaluated in replicated trials prior to selection. And selection for specific combining ability, we will make use of an inbred line, which is generally narrow in terms of genetic base, as a tester. The test cross performance also will be evaluated in replicated field trials. In replicated field trials before selection can be done. Reciprocal. The idea of reciprocal, let me explain something about this reciprocal thing. In reciprocal, let's say you cross parent A and you cross it to parent B. Okay? You have made A the female and you obtained pollen from B. Okay? So progenies that you will make, okay, will be progenies whose mother is A. Now, you can also make A the pollen source. A becomes the pollen source and B now becomes the mother. So you will obtain progenies where there is where B is considered the mother. Okay. In reciprocal selection, you are considering both scenario. You are considering both scenario. You are considering a scenario where A is the mother, and you're also considering a scenario where B is the mother. Okay? Reciprocal. You will see that as we deal with that. Now, this scheme is capable of exploiting both general combining ability and specific combining ability. And it entails heterozygous populations, each serving as a tester. So details we will see when we get to specific types of selections. So I have introduced recurrent parent. I've talked about recurrent selection. I'll post these notes that I require so that you look at them in advance so that we are able to discuss fine details when you are a little more deeply informed. May I stop there to ask if there be questions? Up to that point on recurrent selection. Introduced you to what we are going to talk about when we are discussing breeding for cross-pollinated species. Any questions for me? Remember, as we go deeper, a few things come, so the earlier you ask me, the better. I can see several people are leaving my meeting when I'm not done yet. They may be wrong because I'm continuing. I have just paused a little bit so that we can discuss. Any discussion on this? Any question? Okay. already talked to you about recurrent types of recurrent selection. So now I want to discuss so in that or we can do it through us within a approach selecting population. I have said improvement, which would improvement can be done in several ways. We can do this through selecting individual plants within population, or we can do it through selecting families within that population. So I said when you select single plants or make single plants a unit of selection, we actually practicing what is similar to mass selection that we discussed. But when we do family selection, then we are looking at the various recurrent selection methods. Let me remind you that in individual plant selection, there is a difference between mass selection for line development and mass selection for population improvement. Discussing mass selection for population improvement, the aim is improving a general population mean or population performance by selecting and bulking superior genotypes that are already existing in that population. So what is the key feature of individual plant selection methods? That selection units are usually the individual plants themselves. That selection is solely on phenotypic performance, and that seed from the selected plants are bulked to start the next generation. So really, no crosses are made. Okay, no crosses are made, but progeny test is conducted. And the process is repeated until there is a level of improvement that is desired. So that's the key feature of individual plant selection methods. How is this carried out? Simple. Year one, we plant the source population, and our source population can be a variety. Just like I told you, you can walk to a market, you look at the bean seeds and they don't look uniform, you pick them and you decide to grow them to do mass selection on them. Can be a local variety for a long time and you want to improve it. It can even be a population that has been generated from bulk. Okay? So you bulk them and then begin from there. So the procedure in year one is that they are planted, then you log out what you do not want before flowering. Then you select several hundreds of plants based on the phenotype, harvest then bulk, and then in year two, you grow them out again, and select in a preliminary yield trial, and then in year three, you repeat in what you did in year two, as long as the progress is made, and then you conduct advanced yield trial. So that is the simple procedure for individual plant selection that we know. So I need to request you to take note that mass selection may be longer depending on the progress that you are making. You stop selection so long when you feel you have made enough progress. But what are the genetic issues of this individual plant selection? That it is based on phenotypic trait selection, and therefore will require traits of high irritability, and it is most effective when the gene action is additive, and it depends also on the number of genes that are involved in the trait of interest. The more the additive gene action, the greater the efficiency of mass selection. So the expected genetic advance for mass selection will then be given by a formula that we are seeing here. That this delta G, of course, is genetic gain, will be like a half selection intensity times additive genetic variance, divide that by the total phenotypic variance. What are the advantages of this? This gives us opportunity to break genetic linkage blocks because we are doing what is called repeated intercrossing. It also gives us opportunity. It's applicable for autogamous species like the grasses and the legumes. But the disadvantage is that phenotypic selection makes selection of superior plants difficult, and that lack of pollen control, we are not able to control the pollen, what pollinates what. This brings the desirable and undesirable pollen, which may not will not be advantageous, and that if selection intensity is too high, what will happen is that there's possibility of inbreeding depression. So that is phenotypic selection, individual plant selection. So that is phenotypic selection or individual plant selection. Yes, I can see somebody whose hand is raised. Who is this whose hand was raised? What did you want to say? Okay, so those of you who want to leave my meeting, why are you leaving them? You are not there, of course. Okay, then we have the family selection method, again, which is also characterized by three methods, three steps, that is. There's the creation of the family structure, then evaluation of those families and selection of superior members, and then recombination, of course, mating, so that you use the superior ones that you have selected are mated afresh, so that you generate a new population. But remember that in terms of families, we have family members that can be looked at as half-sib. What is half-sib? It means they are individuals who are sharing, one common parent. That is called half-sibs. They could be sharing a mother, okay, but their fathers are different. So they are half-sibs. In other words, for plants, the pollen source is different, but the mother is the same. Or you have individuals that are full sibs in terms of their relationship. They share both the mother and the father. So here we are going to talk about family selection among half-sibs and family selection among full sibs. In other words, family selection among individual plants that selection plants that share both parents. Okay. So when we talk about family selection, we'll be talking about half-sib family selection methods, and we will be talking about also full-sib family selection methods. And you need to be keen on this. You need to be keen on this. You need to be very keen on this. Like when we talk about ear-to-row selection, what are we talking about? We are talking about half-sib family selection. Or we could be talking about full sibs. And full sibs are generated from biparental crosses using parents from the base population. The families are evaluated in repeated trial to identify and select superior full sib families, which are then recombined to initiate the next cycle of selection. So full sib, half sib stories should be familiar with you and how their procedures are generally carried out. Okay. Then we'll have family selections that are based on test cross, which we will be able to discuss. So we can do a half sib selection with test cross or a half sib selection without test cross. Those are the key things that will be coming out as we do discussion on this particular family selections. Okay, I wish to stop here so that you get opportunity to look at these notes in advance. I will post them. I will look at them again. I remove what I don't want to do there. Then I'll